So thank you, Daniel. I also want to warmly welcome you all to this summer school. My name is Marja Leena Linne, and I come from Tampere University, where I work in the Faculty of Medicine and Health Technology. I used to work in a Faculty of Computer Science and Digital Signal Processing, but nowadays I'm in this very interdisciplinary faculty where I can kind of like do theoretical neuroscience, computational neuroscience, and also experimental neuroscience. And I'm training bachelor, master, and doctoral students in all these three different fields. We typically have this kind of introduction to the summer school in the beginning of this school. I know that for some of you, the things that I'm going to present here is already familiar, so this is okay. But then we also know that there are like those like late phase master students, beginning doctoral students that have very little exposure to this field. So we want to first set the scene, basically, that we talk about the same things, and I also try to give some definitions, and then we can discuss about those definitions later on during the course. So we are all here to understand this system, the brain, that has 3 million kilometers of wire, about 100 billion neurons. And what we not normally talk about, it has also almost the same amount of other types of cells, namely the glial cells. This system is consuming 10 watts energy, which is very little. It is about 1.5 liter in volume. It functions almost 100 years without spare parts, if we are lucky. And the system has incredible skills and is able to learn. So this is the system that we are trying to understand here. So although we know quite a lot about the brain already, we still have many, many big questions in brain research. We would like to understand how the brain functions. And more specifically, we would like to understand what are the genetic, molecular, cellular, and network level mechanisms that make our brain to learn, to remember, and to understand. And this is also kind of the driving force of this course, that we are gradually trying to build you with a kind of theoretical foundation that you would be able to do this type of research in the future. Despite the fact that we know quite a lot about the brain, we still have many, many challenges. And this slide lists some of them. So we are looking really hugely spanning spatial and temporal scales. And we are using many, many different experimental techniques that are illustrated here, from molecular level through cellular and tissue level to whole brain level. And what is important to realize here that rarely a single lab or a single researcher is using all these techniques. So it's like labs are specialized to use a few of these techniques. Some labs already kind of span from molecular level to cellular level and even tissue level. There are presentations later on, for example, Dirk Schubert is kind of spanning through these different levels, a neurobiologist. But what is important to realize that in the future, we all specialize to different techniques, some to experimental techniques, some to more computational, some to theoretical, and we have to collaborate. So another challenge in neuroscience is also that we don't many times talk about, and it's that we use different types of animal preparations, like sea animals, snails, and sea hares. So most of the synaptic plasticity and learning studies at the molecular level have actually been done using these preparations. Then we have traditionally used squid, and all this Hodgkin-Huxley modeling is based on it, that I will come to that later on. And then we frequently now use mouse and rats. But it's different from human. Their brains are different, size is different, functionality is different, and their behavior and kind of goals in life is completely different than ours. So the challenge is, how do we scale between the mouse and the human? And people like myself and many others in our field, we consider that mathematical and computational modeling is absolutely necessary in order to overcome these challenges in the future. So, to overcome these challenges, global neuroinformatics ecosystem was established around year 2000, maybe a bit before. And since then, a lot of this kind of global continent-based neuroinformatics projects have been established. So as you can see from this slide, there are projects in US, in Europe, in Asia, in Japan, China, and so on. Some are more governmental funded, some are like private institutions, like the Allen Brain Institute that is doing wonderful work in bridging different scales and in the brain and kind of like making all data openly accessible. So what neuroinformatics is trying to do is kind of trying to unify all these different fields that you see in this umbrella, different fields of neuroscience. And it's trying to bring the data together in order to facilitate better tools for computational and mathematical modeling of the brain in order to understand the dynamical function of the brain and to help us to cure disease. In Europe, we have the European Neuroinformatics Project, which is a future emerging technology project funded by the European Commission. And this flagship is called Human Brain Project, HBP. Probably all of you have heard about it. It's almost at the end. It started 2013 and will end next year. The goal, many times it's kind of misinterpreted that we solve the mystery of the brain. It's not that. It's more the kind of build the ICT infrastructure to model mouse and human brain and to kind of help facilitating bridging between mouse and human brain. This HBP involves about 20 countries in Europe and I think it's more than 125 institutions right now. So there are those core members that joined in the beginning, and then there were competitive calls that took in new partners every 2 years. The project has 1 billion euros, which about half is from the Commission, European Commission, and the other half is from the member countries. But it's still less than, for example, the similar European US projects, so the funding is less. But in any case, it's the starting point for setting up a digital neuroscience platform in Europe. And we are now in a transition phase. I'm also part of the HBP. So we are now in the transition phase from HBP, the scientific project, to eBrains, which is a non-profit organization to be located in Brussels. And it will provide all these data and tools that we developed during the HBP and continue developing also after this HBP project for brain research. The leader is already selected, is Pavel Svieboda. And what is also important to note that already the Nature journal is recommending submission of datasets to eBrains. So although eBrains is under constant development, it is already becoming like a recognized repository for the neuroscience data. So in the future, this eBrains is supposed to provide the digital neuroscience platform, facilitate brain medicine research, and also brain-inspired technology research in Europe and globally. So eBrains has 5 platforms. The first one is data and knowledge platform. Then there is Atlas platform, brain simulation platform, medical and data analytics platform, and brain-inspired technologies platform. And we consider here mainly this brain simulation platform. So during the tutorials, we get an introduction to many of the tools that are already available in this platform. For example, Neuron, Nest, the virtual brain, and so on. So they can all be accessed also through this eBrains platform. So this data and knowledge platform is generated in such a way that all types of neuroscience data, no matter if you talk about anatomical reconstructions or electrophysiology or brain imaging data from humans, can be uploaded to this system. There is very important phase, this curation phase. So every data that is entered in the system will be curated, and it's kind of like checking up what it is, and like that there is like a sophisticated metadata system behind it, behind the data, and basically kind of validation phase. eBrains also have this called knowledge graph. This is kind of a very center of this system. It operates, in my understanding, the same way as Google has developed their system. So it should facilitate really like a sophisticated system to look for the data in this system, like with the keywords and easy find the type of data you are interested in. And then this data and knowledge platform integrates with all the rest of the platforms, so it integrates to tools and different workflow programs. I will tell you a little bit about these workflows later on. So already now, as I said, a lot of data is shared through eBrains. So the interface looks like this, and here is, for example, a data set where you have physiological data recording of interneurons from human temporal neocortex. This is data already available, and then in some cases you may make like a pre-announcement of the upcoming data and make your Imparco data, so the data will become available when the publication is out. So the system is very flexible to share. And first you can share with your colleagues, then maybe later with the project members, and then on a later phase maybe with everyone on the globe. So medical informatics platform is an interesting platform in the sense that we don't, at least to my understanding, we don't have such a system available on the globe where you could provide a direct link between the clinical and basic brain research data. And with this system, once it's set up and functional, you can share lots of data sets and, for example, test biomarkers and predictive models in clinical settings, for example, in Alzheimer's disease, or discover new findings by increasing sample size of cohorts like they are doing now in traumatic brain injury projects, and facilitate many types of clinical studies where you also try to get some hypotheses from the basic neuroscience data. So this is supposed to be very helpful for the medical doctors in the future. And I think European Commission is investing quite a lot of effort in setting up this system. eBrains also has this brain-inspired technologies platform, as I said, and there is a direct link to, at least now, to Spinnaker and brain scale systems that the HPB is developing. But of course there exist also many other like neuromorphic devices where you can run this type of simulations that we learn here to do on a computer. So in this case you run it on electronics, basically, it's called neuromorphic system. So this eBrains has such a integrated platform as well. And there is also a robotics platform where you can kind of integrate the models, the type of models that we study at this school. You can integrate them in this kind of mouse models that you see on this slide and do robotics research. So this is also a valuable platform for computational neuroscientists. So at this PNNI, we are understanding neurons, cognition and behavior through neuroinformatics and artificial intelligence. So this is in the title that we now expand a bit from the conventional setting of the computational neuroscience, that first you have data on cells, networks, cognition, behavior. You develop mathematical models and then you implement those models on in silico computer models. and simulate those models. But here we also have like talks and exercises related how to apply artificial intelligence techniques. For example, in this kind of case where you have image of a cell culture and try to automatically, with the help of, for example, deep learning, learn like the basic features of these images. So not that the neurobiologist is tracking everything themselves in a very time-consuming manner, but doing this in an automatic way. And then we also discuss at this school how to utilize sophisticated informatics systems like description languages, where you transfer the mathematical model to a kind of like computer-independent description language in order to be able to run your model on any like computer or any tool that is developed to simulate these models. So this is kind of our setting, and I think now, since we are on a 9th year, we are trying to incorporate as much as from this field that relates to computational neuroscience, but at the same time we try to make the lectures in such a way that also those who are in a very beginning phase of your studies can follow most of the lectures and exercises. So I wanted to make also some definitions here. As I said, I want that we all have like the same scene and we start from the same starting point. So computational neuroscience, which is the central theme that we teach at this school, is a scientific discipline that utilizes methods from mathematics, physics and computer science to develop computational in silico models. And then theoretical neuroscience is typically considered to establish theories of brain functions and brain computations, including memory, learning, sensory processing, cognitive functions, and consciousness. Many times we talk about these two terms like as equivalent, but I think they, at least in my mind, they are a bit different, as I explain here. And then neuroinformatics, on the other hand, is an engineering discipline developing tools and formalisms for implementing and analyzing computational models. For example, it develops standardized simulators, data analysis tools, databases, and improves reproducibility of computational studies. So what are mathematical models in neuroscience? And this is the way how I see it. A mathematical model in a neuroscience is a function that describes how the measured response, this YT, depends on the applied stimulus, UT, and on the internal states of the considered neural system, this XT. And these models, sometimes they are simpler, sometimes they are more complex. And the more complex these models are, the more model parameters we typically have. And this makes it very, very challenging field, because if you have a lot of unknown parameters, it's almost impossible to set up the model, or you have to use very sophisticated data fitting procedures in order to find out those unknown parameters. But luckily we have a lot of data nowadays, and we can kind of we know many of these parameters of the systems that we model. So you will learn this type of models at this course and how to implement them. In the very early phase of the computational neuroscience, when it started in 1990s, one scientist typically worked only on one level, so maybe on a neuron or network circuit level. But nowadays, almost all of us are kind of reaching at least 2 levels, or maybe the whole span of these levels, like from genes up to whole brain when we do modeling. And quite many models on each of these levels already exist, as you see from this slide. But the challenge is getting to kind of develop an expertise to see which model to choose when you do modeling on a different phenomena, and ask specific questions that which is the best model to choose. So that is the challenge of computational neuroscience. So a typical neuroscience workflow, and this is exactly what this e-brain system is trying to develop, that we make this kind of a more conceptualized, that there are these workflows that everyone follows, and kind of like reports when they do science, reports these steps like in the same way, so that it would become science in neuroscience would become more replicative and reproducible. So there is first of all this neurobiology, where the neurobiologists obtain all data. Then they store it in data and databases in an ideal case, and do data analysis with tools that are validated, and then kind of feed in to the modeling community who set up the models and do the simulations and computations, and then the visualizations of the data. And the idea in the future is that we have kind of like standardized, well, not maybe standardized, but at least semi-standardized systems when we report our results, and we do it in these kind of workflows. If we look at the modeling workflow in more detail, we see that there are also many steps there. I see myself that there is first a step where you do the problem statement. So you, for example, get a specific question from your neurobiology collaborator, like how does certain mechanism influence the behavior of this animal or the behavior of this cell culture or something like this. And then a computational or theoretical neuroscientist describes a model to explore this specific question. And then follows the model specification, where you really have to find data for the parameters of your models, or at least have your educated guesses what are those parameters. And then maybe like later on, once the model is implemented on a computer, then do like automated parameter fitting. And the idea is that in the future, in this workflow, all these steps are kind of like automatically reported in such a way that they are like computer understandable. Then there are these model analysis phases, and importantly the validation phase, where you are supposed to kind of validate your model output against the experimental data, which is different than you used for setting up the model. And then, if you end up with a model that is good enough, you are lucky to make predictions about the neurobiological phenomena that you are studying with the in silico model. And in the most lucky case, the experimentalists later verify these predictions, and then it can be concluded that the model is really like a predictive or have predictive capability. For those of you who are not the experts in modeling, I wanted to give a little bit guideline, like a really small, simple project in computational neuroscience. If you are skilled in software, may take a few months, but typically a large-scale model that you develop in collaboration with the neuroscientists, it takes years, basically. So the question is now also like, in the field of neuroinformatics, and when we are applying this engineering, neuroinformatics, AI technology, is that we would like to speed up this process. We would like to make more replicative, reproducible models that can be more easily hooked up in a new project, and kind of like speed up this process to develop models for different brain areas and different brain cells and so on. And this is why we would like to set up these workflows to make these modeling projects more understandable for an outside person. So let's look at the classification of modeling approaches next. Sometimes models are classified according to the complexity of the model. So you may have like these detailed mechanistic biophysical models, where you start from the details of the cells and then go on, as illustrated in this figure, to describe the networks and even the kind of like populations of cells, where you don't treat the individual cells anymore, but like look at the populations. But you have started the from the kind of like biological details. And an example of these population level models are the current state of the art mean field models or mass models, how they are called, which are kind of like biophysically informed, doesn't have all the biophysics anymore there, but kind of dynamically follow the biophysics. And then we have this other type of models that we call sometimes simplified or conceptual or phenomenological models, if you look at the literature. And there you start from kind of like going from top down. So you try to explain some phenomena just with a very, for example, with a statistical model without kind of forgetting the biophysical details. So these are kind of two different ways of seeing the modeling. And then many times we also talk about like either bottom-up models, which are from mechanisms to behavior, or top-down models, which is more the models from behavioral targets to mechanisms. So when we talk about computational neuroscience, there are two fundamental CNS models that should be mentioned here. And you will have an exposure to these models during the course, so during the first and second day. So there will be Dieter Jäger and Ann Rood, and also Hermann Kunz, who will be kind of like discussing the theoretical foundations of these models. And these are the model of Hodgkin-Huxley, which is a mathematical model for action potential initiation and propagation. It's basically a set of nonlinear differential equations that you see on the slide to produce the action potential. And then there is the RAL model, which is, in my mind, a kind of like biophysical mathematical model of neurons with dendritic trees without any activity, more the geometry of the dendrites. And it's called also Capel model. And as is shown in this slide, we kind of merged these two approaches that were developed very early, actually like in 1950s and 1960s, together in order nowadays to model very large compartmentalized neurons of the brain, like the pyramidal cell of the cerebellar cortex and the Purkinje cell of the cerebellum, with this approach. And this is something that you will also apply at this course, these techniques. And it's important to realize that also dramatic increase in computer power has occurred since 2000 or so, and we have had like lots of new models in this field. So there is history, and you have had all these theoretical foundations developed early on. There are also other neuron model types, not only the biophysical models, as you see from this listing. So the higher upper you go in this list, you see there are more and more kind of like phenomenological models. So integrate and fire model and its derivatives, you will, I think, learn during the second day when we have the next tutorials and lectures by Sassavan Alpada and others. And there is also like one model that I illustrate here, the Ichikevich model, which is purely mathematical model, but as you see, it has these pluses, so it's almost reproducing all these behaviors of neurons in a mathematical way. But of course, Hodgkin-Huxley model is the most biophysical one, and is able to do most of these neuronal outputs that the neurobiologists measure in their experiments. But as already this table shows, there is already, when modeling neurons, there are many like models to choose from. And it always depends on the research question that you are asking for, which one of these is useful to choose. And then also depends like what you choose on your computing power that you have available. If you can use supercomputers for your task, then you can choose maybe Hodgkin-Huxley, but if you have more limited computing power and would like to describe large networks, then you have to be a bit critical like what to choose here. So how do these computational neuroscience models help? So based on this description I've given to you, maybe it sounds like very theoretical. We try to implement something and try to explain some aspects of the brain. So how does it help us? So I see, like, at least when I started this field already in the early 1990s, I immediately saw that this is the way to make sense out of experimental data. Maybe this is because I'm originally an engineer, not a neuroscientist, and have gradually become a neuroscientist later on. So these models really help to integrate the data and put it in a meaningful perspective. For me, somehow it's too complex to read the textbooks and see all these little details. But once I see a model and things are put together, like these dependencies of different mechanisms, it's very intuitive, at least for me. Then secondly, these models, they describe neural systems and they also fit existing data. But what is most important and what we aim with these models when they really become sophisticated is that we could make predictions and to help the neurobiologists to kind of maybe allow them to kind of reduce maybe the experiments and redo something in silico actually. And then they can make a smart plan what type of experiments to do. But I don't think in the future computerized simulations will never completely replace the experimental work, but they can help to reduce the amount of experiments that are needed. And of course in the future, and like we saw at the end of my presentation, there is really hopes that we could kind of like provide mechanistic understanding of the brain and also help to develop new tools to treat brain diseases. And of course, we are already now, there are serious attempts like interfacing the neuroscience and brain research with the artificial intelligence, developing new technology, more flexible systems than we have currently, for example, in deep learning and AI field. So with all these data, all these models and all these tools, we have been able to develop a few quite large-scale models of different brain areas. So there is a model for cerebral cortex, cerebellum, hippocampus and basal ganglia, I think. And here I took two examples. So there is this model for mouse somatosensory cortex, which is perhaps, at least in my understanding, the most detailed existing microcircuit model of the brain of the cerebral cortex. And it has this 6-layering structure, this model that is known to exist in the cerebral cortex. It has measurements from rodents, but now we can also obtain data from human patients, epilepsy patients, for example, to improve this model more towards the human type. And with this model, one is able to simulate spontaneous activity in this somatosensory cortex of a mouse in vivo and look at the spontaneous brain state changes. And one has like short-term plasticity and such there, but it's important to realize that we are not able to kind of model the long-term changes in the brain and all the memory processes. So that is still missing in these models. So this gives you an idea where we are now. But this is a very good starting point to start exploring the in vivo system. Then we have a similar model for cerebellar cortex, which has these 3 layers, as maybe some of you know. And there is a very good reconstruction of this anatomy and also the physiology. You can look at this paper, for example, Casali et al. In Frontiers in Neuroinformatics. And then there is a model about the cerebellum where you can explore these dynamical state changes between different states that this cerebellum is expressing. And both of these models are relatively well validated against experimental data measured from rodents. And this is something kind of a new direction. So these 2 studies and these 2 models kind of, at least to me, they show the future, how we are supposed to kind of go on. But as I mentioned, there are important phenomena and mechanisms in the brain that is still mostly missing in our models. It's the homeostatic regulatory processes, plasticity processes, learning and memory. Some models have some model are modeling these phenomena, but most of the models still don't have it very realistic. And we currently know, as you see from this slide, that it's very complex, because we know that plasticity and learning, it's not only the cell membrane phenomena or network phenomena, but it's also dependent on what happens on the cellular signaling level and even in genes. When you think about long-term effects and like recruiting new AMPA and NMDA receptors, for example, on the cell membrane. So for this, you need the molecular and genetic machinery to be in place. And how to model this, because in these really brain level, full brain level models, you cannot model every single molecular movement or diffusion of ions or molecules. So it's big questions in our field how to do this modeling. There are, however, already attempts to this direction, and there is, for example, this study by Zenke et al. Friedman Zenke is also going to give lectures at this school, and based on the synaptic plasticity and learning data, biology-inspired learning rules have been developed in this study. There are 3 different synaptic plasticity models that have been incorporated in this spiking recurrent network model that has both excitatory and inhibitory neurons. And with this kind of model, the authors were able to provide sufficient cell assembly formation and memory recall in this kind of more phenomenological brain-inspired network modeling approach. And you will hear more about this type of studies as well, where the biology-inspired systems from the computational neuroscience field have been incorporated in the artificial neural network studies. And then on top of all this complexity that we have in the brain, there is the complexity of glial cells, which this field and neuroscience in general has completely neglected. So here is an illustration of the cerebellar Purkinje cell. We've thought that it's the biggest cell and most dense cell in the brain, but then we see that the glial cells, they are actually, when we now are able to reconstruct them, they are more more dense than any of the cells in our brain, at least the cortical glial cells. And this is also something we cover a little bit in this school, and I'm going to talk about this, modeling these glial cells on Wednesday. So how much time do I have? Do I have time for the applications, or should I stop here? Yes.
It's 2 to 10, and then we have the coffee break. And
Okay.
it would be nice to have you ask some questions.
Okay. So let me just quickly run through the 4 slides to show the applications on a title, and then so now we've kind of discussed about the theoretical and computational neuroscience and all these models that exist there and what we need to learn and develop in the future. But it would be important for you to also understand where to apply these and what has been done already. So in basic science, an example could be that you do computational neuropsychiatry. You go from genes up to understanding schizophrenia, like in this study. And I will share these slides with you so you can look at this in more detail later. Then you can do clinical applications and use these models in personalized epilepsy treatment. And you use these mean field models that I mentioned. And you run through all this pipeline to make personalized epilepsy treatment for those patients that, for example, have rare epilepsy disease that they cannot take any drugs or don't get any help from them. And then there are biomedical applications like development of prostheses for blind patients where you use these computational models, and engineering applications. So this was a quick run through the course.
Thank you very
And
much.
at the end, some literature for
Thank
you
you very
to
much,
study.
Marja Leena. So now you have coffee breaks, but before that, there's two things. So one thing is, maybe we have some questions from the audience. I should say, actually, we have some people online. So guys online, I hope that you are hearing me. If you have questions, you can ask them on chat, or you can just unmute your microphone and ask them. I mean, I don't know if the unmuting works. If it doesn't, just write on chat. So do you have any questions within the room? Or yes.
Thank you for the presentation. When you were describing the usual computational neuroscience workflow, you put the predictions at the end. Usually the predictions in other fields of science come way before, so I assume it would be around model specification. What's the reason for this kind of?
Yeah, of course, they could be part of model specification as well, but somehow I don't know why we present it always at the end. Maybe it's the reason that typically how the model development in computational neuroscience happens is that the first model for certain phenomena is many times not the best one. I think there are cases where the first model has actually been the best one. Maybe the cerebellar Purkinje cell is one of those examples. But in many cases we kind of do the gradual development. So then new research groups take the model and starts further developing it, and it's through many iterations maybe the 10th model is the one that will be done from that model category to do predictions. So maybe it's for this reason is at the end. But maybe like if you think about control theory and other fields where you also like physics where you develop models, so it is also done earlier. But what I didn't mention in this presentation and what is always pointed out by Kaute Ainevoll, for example, that what makes our field very unique, and this is very important to realize. In physics, we can kind of like make models that like statistical models where you kind of like look at the average behavior of, for example, molecules or gas or something, gas molecules. But in neuroscience, in principle, and the neurobiologists should correct me, but in principle, a single molecule could make a difference. And this makes our field very challenging. How to develop models if such a possibility occurs that a single molecule or a few molecules, 3 molecules, can make a change in the dynamics of the brain. So this is why it is very challenging, and model development is iterative, and many times it's the 10th or 20th model developed for certain phenomena that is predictive enough. So maybe that's why it's at the end. But it's a good point. Maybe I should reconsider my workflow.
Thank you. And then the validity of this model is instead through this iterative process, and not whether the predictions of the model would fit the actual observations.
I know I didn't get like what was your question.
No, I was just testing my understanding that the validity of the model would then be decided on this within this iterative process instead of whether the predictions of the model would.
Yeah, it's a good point. Of course, the model is also validated. You have kind of like in between these kind of checks. Many times, like you develop the model in parts and you integrate it at the end. Like the kind of synapse model I have developed with Ausra. So we first developed the post-synaptic and presynaptic terminals, and then the glial model. So the kind of tripartite synapse model, and then you integrate it at the end. So of course we have validity checks also in between. So it's a good point. I should actually mention it on the slide. Yes.